The endoplasmic reticulum is a network of membranes found throughout the cytoplasm of eukaryotic cells. It is involved in a variety of functions, including protein synthesis, protein folding, lipid synthesis, and calcium storage. The endoplasmic reticulum is a multifunctional organelle with diverse roles in eukaryotic cells. Function of endoplasmic reticulum: protein synthesis and processing. The REFA, studded with ribosomes, is the primary site for protein synthesis. Ribosomes translate mRNA into proteins, which then enter the lumen. Inside the proteins undergo folding, modification, for example, glycosylation, and quality control. Misfolded proteins are identified and targeted for degradation. VIA also transports proteins to other organelles or the cell membrane. Lipid synthesis: the smoothie is responsible for synthesizing lipids, including phospholipids, cholesterol, and steroids. These lipids are essential components of cell membranes and are used for various cellular functions. Calcium storage and release: VIA, particularly the smoothie, acts as a major calcium storage site. It sequesters calcium ions and releases them in response to cellular signals. Calcium ions play crucial roles in various cellular processes, including muscle contraction, signal transduction, and neurotransmitter release. Detoxification: the smoothie contains enzymes that detoxify drugs, alcohol, and other harmful substances. These enzymes modify these substances, making them less toxic and easier to eliminate from the cell. Carbohydrate metabolism: the smoothie is involved in carbohydrate metabolism, including the synthesis and breakdown of glycogen. Membrane synthesis and transport: VIA synthesizes membrane components and transports them to other organelles, such as the Golgi apparatus and the cell membrane. The endoplasmic reticulum is a complex network of membranes with a distinctive structure that supports its diverse functions. Two main types: REFA, characterized by ribosomes attached to its cytosolic surface, giving it a rough appearance. The ribosomes are responsible for protein synthesis. The REFA is often continuous with a nuclear envelope. SMOOTHA lacks ribosomes and has a smoother appearance. It is involved in lipid synthesis, carbohydrate metabolism, and detoxification. The SMOOTHA is more tubular in shape compared to the REFA. Lumen: the space enclosed by the membranes is called the lumen or cisternal space. The lumen is a distinct compartment from the cytoplasm and provides an environment for protein folding, modification, and transport. Membrane composition: the membrane is primarily composed of phospholipids, similar to the cell membrane. It also contains various proteins involved in protein synthesis, lipid synthesis, and transport. Dynamic structure: VIA is a dynamic structure that can change its shape and organization in response to cellular needs. It can expand or contract, and its different regions can interact and communicate with each other. Organization: VIA is often organized into distinct domains or regions, such as the REFA, SMOOTHA, and transitional involved in vesicle budding. These domains have specialized functions and are interconnected to facilitate communication and transport. In essence, VIA structure, with its interconnected membrane network, lumen, and specialized domains, allows it to perform its diverse functions in protein synthesis, lipid synthesis, calcium storage, and other essential cellular processes. Now we see the main difference between rough and smooth endoplasmic reticulum. Difference: rough endoplasmic reticulum RER asterisk structure. The RER is characterized by its studded appearance, with ribosomes attached to its surface. These ribosomes give the RER its rough appearance under a microscope. The RER consists of interconnected sacs and tubules. Asterisk function: asterisk protein synthesis. The primary function of the RER is protein synthesis. Ribosomes on the RER synthesize proteins that are destined for various locations, including secretion outside the cell. incorporation into the cell membrane, or transport to other organelles like the Golgi apparatus or lysosomes. Protein modification and folding. The RIR also modifies and folds newly synthesized proteins. It adds carbohydrate groups glycosylation to proteins, ensuring proper folding and function. Protein transport. The RER transports proteins to other parts of the cell via vesicles. Location. The RER is abundant in cells that are actively involved in protein synthesis and secretion, such as pancreatic cells, which produce digestive enzymes, and plasma cells, which produce antibodies. Smooth endoplasmic reticulum here. Structure. The SER lacks ribosomes on its surface, giving it a smooth appearance. It consists of a network of tubules and vesicles. Function. Lipid synthesis. The SER is involved in the synthesis of lipids, including phospholipids, cholesterol, and steroids. These lipids are essential for cell membrane structure and function. Carbohydrate metabolism. The SER plays a role in carbohydrate metabolism, such as the breakdown of glycogen into glucose in liver cells. Calcium storage. The SER stores calcium ions, which are essential for various cellular processes, including muscle contraction and signal transduction. Detoxification. The SER detoxifies harmful substances, such as drugs and alcohol, by modifying them to make them less toxic. This process is particularly important in liver cells. Location. The SER is abundant in cells involved in lipid synthesis, detoxification, and calcium storage. Examples include liver cells hepatocytes, cells in the gonads which produce steroid hormones, and muscle cells where the SER is called the sarcoplasmic reticulum. Thank you for listening. Don't forget to like and subscribe my channel for